We have presentations from other research infrastructures. I think the first one is from Jose Alonso, that is going to present the DISSCO research infrastructure. Did you upload your presentation? I'm sorry? Yes. Okay, very good. So could you make it full screen and make sure that it is, they see it from... Yes, so let me, 2 words of presentation. Okay. Gabriele, do you want to present? Yes. From here, from there. From there. Hello, everybody. Can you hear me? My name is Gabriele Bucci. I work at the National Research Council of Italy, the Institute of Biosciences and Bioresources. And today I will co-chair the session of the presentation of the different research infrastructures that have been involved in this. Sorry? Louder. Okay. Okay, a couple of words of considerations about why we involve the other research infrastructures, and these in particular research infrastructures, which are DISSCO, ELIXIR, METROFOOD, LifeWatch, Emphasis, and MIRI. Today we will have only 4 representing 4 persons representing these research infrastructures. We start with the DISSCO, as well as the other 5 research infrastructures are included in the ESFRI roadmaps, and we found that they act, most of them act in complementary fields, looking at as compared with the PGR research. And so today we have Dr. Jose Alonso from the Naturalis Research Center in Leiden, Netherlands, right? Okay, DISSCO is from the domain environment, entered the ESFRI roadmap in the 2018, and the operation are expected to start in the 2025, right? Okay, hopefully. Okay, the statutory seat is in Netherlands, and currently involves 12 countries as far as I know, but is expected to include at the end 23 countries. DISSCO aims to digitally unify all European natural science assets under common access, creation, policies, and practices. And the main focus, Jose, is on natural history museums, right? And botanical gardens. It is expected to collect different kind of information for 1.5 billion specimens. And it's currently in its preparation phase. Okay, Jose Alonso, the floor is yours.
Let's see. I don't think that the louder will be a problem with me. Yeah. Yeah, sure. I'll try to keep it short. I'll try to keep it engaging. That's what we do, right? Well, thank you, first of all, to Mike for hosting this. It's a real pleasure, not only for being here with you, but also for being here in Greece. I'm from South Spain, so it's like feeling at home, basically, you know, in terms of weather, plant varieties, and etc. So it's really, really a good opportunity to present DISSCO here. Well, Gabriele said it before, we are research infrastructure in the making. That's important, you know. Communication about research infrastructures isn't always an exercise in managing expectations. So we're a research infrastructure, a future research infrastructure. That's important to say. Anyway, let's go. Oh, there's a number of a number of people wanting to enter the Zoom. I can do it for you. It's fine. I guess. Yeah. No. Maybe I need to do that.
Yeah. Nació.
All right. So I am not completely 100% sure that we invited the people who were in the waiting room. I'm just going to assume that we did, hopefully for them. Yeah, I like an audience anyway. So, as I said, research infrastructure in the making. I'm going to try and be very quick at the beginning of this presentation because Giovanni has said it all. We have a number of problems in this planet, right? Most of them are big enough to actually threaten our way of life, right? So as we say in this slide, current global challenges demand that we scale up and speed up biodiversity research. If we continue doing things the way we have been doing them for the last, I don't know, 200-300 years, it's going to take decades for us to gather the knowledge that we need, to tackle these threats. So we need a new system. We need a new. There you go. Some people are still waiting. I think it's important that we let them in. Can we actually let them in? I mean, it's only fair. Okay, that's good. Well, we need, as I said, to scale up. We need more science, more people involved, and we need to speed up. We need to speed the processes in biodiversity research. We believe in DISSCO that natural science collections have a role to play in this. We provide same way that probably Progress will in the future, a humongous base of primary reference data about geo and biodiversity that other research infrastructures can use. But we're not doing perfectly anything. Basically, we need to change a number of things, right? We need to embrace, first of all, I'm talking always about natural history collections, natural science collections. We need to embrace new developments, most of them having to do with data, let's say. One of them is digitization. Today, less than 10% of the specimens in Europe, which is like close to 2 billion with a B, are less than 10% are digitized. It's not much, obviously. We will never be able to work together in tackling these big global challenges unless we have something to work with. And for that, we need to digitize stuff. We need to embrace new forms of data, for instance, genomics, which is bringing new information that we didn't know before. We don't have in tags in the physical specimens in collections. And then also we need to integrate better, and we need to be able to interoperate better among institutions. We always have this running joke at DISSCO, and the running joke says that washing machines are already doing this thing. You can actually deal with them online. So they're embracing new developments. And no matter what kind of model you have, your plates, your glasses, everything always fit. So obviously they interoperate well. I mean, they're providing this information. We believe that we have better reasons to actually work on this because let's say that the future of the planet depends on what we do, right, in science. So yes, these are the two, let's say, big challenges, so to speak, and that's a reason d'etre of DISSCO, right? Gabriele said that before. DISSCO brings together a number of European natural science collections into a digital infrastructure. Has to be digital, of course. We cannot bring them together in a big building because there's no such building. Has to be digital. That will function as a unified collection for all of Europe. That unified collections is meant to follow a business model where all the collections share curation, access, policies, protocols, processes across countries. That's the goal. That's the goal that we're trying to pursue. Today, as in October 2023, we are 178 institutions already being partnered of DISSCO across 23 countries. What's the value proposition of ours? Well, what we plan to do is we want to create a one-stop digital infrastructure, otherwise called a single access point, if you want, where we will provide both physical, it's not about forgetting about physical access to collections, of course, we will provide physical and virtual access to natural science collections. Data, expertise, and facilities. I'm including the term FAIR. I'm not completely sure how familiar we are with FAIR. FAIR stands for findable, accessible, interoperable, and reusable data. It's a set of principles that are meant to have data open and accessible for all kinds of uses in research. We will get back to that anyway in a few minutes. So one digital infrastructure that provides that kind of access, and that provides a number of user services, so we can do better things with that data that we now will have, hopefully, accessible in due time. Those services have to do with digitization on demand, of course. As I said before, less than 10% of the data is digitized. We need more. Will have to do with enhanced interpretation. We're talking about image recognition. We're talking about research support, of course. We're talking about training, etc. So one single access point with a lot of data in it, to put it very simply, and with a lot of services in it, so we can actually make the best out of that data. We like to, it's a little arrogant to say this, okay, but we want to talk about a digital transformation of whatever is happening today in the landscape of natural science collection. Let's take a look at that model. On the left, you have the current situation. Eva Solves. Okay, I understand that somebody letting people in, right? Well, okay, that's right. Thank you, Panos. Current model is the one that we basically live in today. You're a scientist. You're interested in one specimen, one type of butterfly, one type of beetle, one type of whatever. You need to travel. You need to request loans and visits. You can only have access to a number of physical facilities. It's slow normally. It is expensive, not only in terms of money, also in terms of carbon footprint. And the one on the right is the one that we want to build. Again, we want to build. It's not done yet, right? This is important. I'm saying this because I'm normally, my colleagues normally say, okay, you always say that things are already done. No, they're not done. Okay, we're working on it. This is very important. An integrated research infrastructure model in which the access is global through that single access point that I explained before. Lower costs, obviously, you don't have to travel. Less carbon footprint, of course, because you don't have to travel. It's faster and is ready to host fair data. Again, we'll get back to that later. And artificial intelligence methods. That's also really, really important. We said before, scale up, more science, speed up. Speed up has a lot to do with methodologies of artificial intelligence. Machines do things faster than we do. So we need to have them do whatever they can do, right? All right. Now, how are we gonna do the model on the right possible? Well, there you go. We have a proposal, let's say, right? DISSCO technical architecture. What we want to do in this while we're developing this digital architecture is to transform what today are static records. You go to a natural history museum, you see a lot of specimens with a tag on it. That information there is fixed. There's no way to change it, basically. No ways, but let me get the metaphor right anyway. So there's a tag with something written on it, nothing to do with that, other than reading. We want to transform that into dynamic, actionable digital objects in an integrated landscape, the one that I showed before, right? How are we going to be doing that? Well, we have a bet. We call it the digital extended specimen. This is very important to underline too, to make it on the record. The digital extended specimen, of which I'm going to be talking about right now, is not exclusive of DISSCO. We have been for some time here in Europe developing the concept of digital specimen, but it so happened that at the other side of the Atlantic in the US, they were developing the extended specimen. So once we realized that, we got together and say, hey, how about we work together, right? And let's call this thing digital extended specimen. So it's been developed in collaboration with other institutions in the United States and other countries as well. Usually, we define the digital extended specimen as a copy, as a surrogate of the physical specimen. You have that little super cool beetle in there that it's laying on some collections, physical, on the shelves of some collections. And we have a digital copy, a digital twin, we like to call it. But it's not just a representation visual like you can see in this slide, obviously. It's dynamic and it's actionable. It is dynamic because even though it is just single unit, single package of data. It can link to other FAIR data, findable, accessible, interpretable, reusable. You can see that there under the screen. It can link to other FAIR data about the same specimen in other locations, in other institutions. This is very important. We don't host the data. Otherwise, we would need, I don't know what kind of drive, right? But we link that package of data to other places, to other institutions that have also information about the same specimen. So that would be the dynamic part of it, because we will continue integrating new information about that same specimen. It's not the tag anymore. It's the tag plus, obviously, right? And then it's actionable. In other words, it can be curated, it can be annotated, and this is very important, by humans, as always, but also by artificial intelligence systems, by machines. We'll get into that a little later. Let's talk a little bit about the anatomy of the digital specimen. I'll try to go fast in this because it's a little complicated, and to be completely honest, I am not the technical guru or anything, so I'm just the outreach person of DISSCO. So please don't have any technical questions from me, I beg you, from now. Part of the digital specimen, one of probably the most important part of the digital specimen is the persistent identifier. We use, among others, you might be familiar with an ISBN for books, of ORCID, if you're a researcher. Those are identifiers, and they have a number of characteristics. They're searchable, searchable. They're resolvable. You click on them, you go somewhere, you know, and they remain unaltered across time, right? So each digital specimen has a persistent identifier. In this case, we have a DOI from the DOI Foundation because it's simply the most, I mean, we understand that it's the best for our case, right? The good thing about the persistent identifier is that it ensures the uniqueness of the digital specimen. No other specimen in the world in history will have the same identifier as. It's like a passport number for the specimen that will never change. Never say never, but you know what I mean. Because it never changes, it will contribute to long-term trust in the data because it will be there. It will be curated by a number of institutions. We will see that later. So we will have, let's say, better or more reliable information about the specimen. And it puts the, well, I meant to say the F, not the FA, on FAIR. It's findable. It's just a code. Same way we use HTTP, blah, 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 blah. You can actually say DOI.1000/ blah, 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 and you will get directly to this thing, to the information on this digital specimen. What else does it have? Obviously, the single package of data has information about the specimen, the tag, let's put it that way, you know. So it connects with the actual physical specimen that is hosted in a certain collection, and obviously facilitates the possibilities of loans and visits and permits and all that kind of thing, because all the information about the physical specimen is there. What else does it have? What we call derived information and associated information directly from the specimen or associated with the specimen. We're talking about genomic information, morphological information, ecological information, literature, biochemical, all kinds of data. Links to that data, to those kinds of data, are included also in the package. And of course, we have media, we have photos, we have scans, we have graphs, we have all kinds of things that are visual, right? As I said, specimens, a single package of data that links to a lot of other information. Now, if we need to position this development, 3 minutes, okay, fine. We will see that there is a fundamental transformation. You know, it says here, you can read actually, the digital specimen aims to allow DISSCO change the model from one model, the one more or less today, in which individual taxonomists curate static specimen information in individual institutions, to one that is, we saw before, dynamic, that is actionable, and that is integrated. It's integrated in an integrated model. Sorry, I mean, I got lost in my own explanation. And that also implements artificial intelligence mechanisms. So we have dynamic information, curable and annotable, if that is a word, information that is inside an integrated system, and that allows for speeding up things with artificial intelligence. Oh, let me just go back a little bit. This is the more or less the model that we have today. You have an individual content management system that is a collection. You have individual taxonomies, they curate, they annotate, and then that data can be served to a data aggregator. GBIF might be something familiar for most of you here, GeoCase in the case of geology, and those obviously reach out to researchers. The model that we kind of predicate here, that we sell here, is one in which not only one, but multiple institutions, content management systems, are linked to the digital specimen, a model in which the community, not just one, but the community can annotate and can curate this dynamic package of information, which is the digital specimen. Also, artificial intelligence systems can actually annotate and curate the specimen. I provide a few examples in there. Image recognition is something that we're planning to do with artificial intelligence. Quality of the data, fixed dates, fixed taxonomy, mistakes, errors, whatever. So those kinds of things are the kinds of things that machines can do for us. They will do it in the digital specimen. And then all that package of data, it can actually go to the data aggregators and ultimately to the research community. Now, is the digital extended specimen going to solve all the problems that we have in interoperability? Absolutely not, of course, because as you know, it depends on many other things, some of them technical, some of them not. But at least we will facilitate the connection among institutions, the connection among curators and researchers, etc. We will contribute to the harmonization of data. The moment that we have a persistent identifier, people will be able to locate the information the moment they want it immediately, let's say. And we will also increase the verification, the openness, if you want to call it that way, of the data. So in a way, that relates into, or that translates into more interoperability around institutions. Now, the digital extended specimen is something that is being, I'm going very fast now, Gabriele, I'm seeing your face. It's something that is being developed within DISSCO, but also applied in other projects, other future research infrastructures. One of them is Bicycle, and more importantly, probably for you, BioDT, which stands for Biodiversity Digital Twin. They are developing that one. They have one use case, which is related to crop wild relatives. And now, as I said before, and yeah, just like one more minute and I'm done, promise. We are doing this thing. I mean, we're not done yet, okay? We have had a very long preparation phase in which we were trying to get the implementation readiness level in terms of data, technical matters, financial, organizational, governance. We have been working on all those things to be ready to be able to construct. What happens? COVID happened. So now we have a DISSCO transition in which we will be able to work together again after the long pause because of that thing, and we will be able to consolidate technically the services that we want to provide, and also in terms of organizational matters. We want to become an ERIC. Giovanni mentioned it before. It's not the only option, but it's the option that we believe is more interesting for us. So during this year and a half, 2024 until 2025, more or less, we will be working on that to get ready to start the construction phase in 2025 to have things done, let's say, around 2026, all right? I have a number of slides about individual services here. I guess that this presentation will be ready for everyone who wants to take a look at it. So one of them is a unified curation annotation system. We have seen before how we will, or in principle, we aim to allow a number of annotations and curations from the community. That's how, with these services, we will have a single access point for loans and visits. So you just have to, rather than dealing with 23 countries and I don't know how many institutions and their own ways of doing requests and loans, etc., we will have a unified access for that, which is also already in beta version. And we will have things like the digitization dashboard. We call it that, collection digitization dashboard, in which we can compare collections. Let's say that you're interested in one type of butterfly. Okay, let's see what collection has more of them. Let's see what kind of preservation methods they're using for that butterfly, for that beetle, for that whatever, right? That's the thing. Number of next steps to take, all of them technical. Full disclosure here, I don't understand more than half of that slide in there. So as I said, please don't do any technical questions now, and thank you. I think we have please do.
Thank you. Thank you for a very interesting presentation. Now I'm going to ask you a question.
There you go. We're going to have to wait. Thank you.
also for the people online, all the questions will be postponed to the roundtable that we have just after this session. So the next, I was told that I was too long and wasting time. So I'll be very short in presenting the second research infrastructure, which is...
Morning. So I'm Roland Pieruschka, responsible for the coordination of emphasis. I'm from the research center in Jülich, and yeah, we are working on emphasis in a similar way as Alonso described in DISSCO, so doing the same S3 roadmap and developing emphasis as a future research infrastructure. So the basic idea to start with a general introduction, why do we do plant phenotyping? So phenotyping, obviously, we want to understand the interaction between genes and environment, which shapes the phenotype, which is relevant for plant quality, quantity of biomass, novel characteristics, products, yielding in stressful environments, sustainable production. So basically all those different plant traits that contribute to plant productivity. So breeders were already addressed by Giovanni. So this is the aspect where breeders basically need this phenotypic characterization, which is needed more in the end. Knowing just the DNA sequence is by far not sufficient to go into a breeding step, and you need also to know the phenotypic traits of a plant to really make progress. So, oh, I try to get closer. Okay, is it better? So demand for plant phenotyping. So the general goal is towards sustainable plant production, sustainable agriculture. In principle, what we need phenotyping, quantitative assessment of specific traits, which needs to be assessed combined with technology. So we go into really high throughput. So without specific technology, it does not work. It cannot be done manually. And in the end, we will never be able to screen all the traits in experimental way. So there is also a need for systems for multi-scale modeling. In more details, so what we need, the tool, plant phenotyping, and we are always talking about the tool. It's a tool to do research, to address different questions, and these questions means crops, conditions, and therefore we need all the specialized infrastructure to characterize plants, to characterize the environment, simulate the environment. Then obviously expertise is needed to analyze pipelines, modeling, data reusability, integrated multidisciplinary approach. This is pretty straightforward. So we have a very wide community with many different expertises, and Europe is a global leader. This is one of the aspects of S3, but there is a competition growing, and we need to work on those questions in specifically addressing agriculture jointly within across countries in Europe. This has been recognized by many different countries. So, you can see that there is many national infrastructures in Germany. This is in Austria, in France, in Ireland, in Netherlands. So in many different countries built their national research infrastructures. So quite often they spend a substantial amount of money, 10, 20, 30, in some countries 40 million euro, to build infrastructure, to have these phenotyping facilities and provide them for access, for use, for the user community in their national level. And integration across the countries happened in many different projects. There is a number of those projects. I probably will not go into the details. Some of those projects addressed access across borders. So this transnational access, which you may be familiar with. But there is obviously a disadvantage. This is on a national level, regional limitation. On the project level, obviously there is a time limitation. After a certain period, the projects end, and basically you cannot proceed. Therefore, we applied for emphasis as a long-term stable organization to provide, to bundle, to integrate all those services and provide also a long-term perspective for the user community. So emphasis is listed on the S3 roadmap since 2016, and we are in a process of also becoming an ERIC. Very briefly, the road to operation, there is a number of kind of lessons learned, but it probably would take too much time to discuss that now. Happy to discuss that also afterwards within the next days. But very briefly, so we had a slightly different process than you have with Grace. We started with a preparatory phase. So there was an application and immediately a preparatory phase where we developed basically a business plan, how Emphasis could operate. With you discussed about different stakeholders. So basically mapping what is the infrastructure, what are the stakeholders, what is available, what is needed by the community. And this was done within the first period with development of a first business plan. The final goal is to have a legal framework in place. Eric, there are many different, so Alonso mentioned this, there are many different legal frameworks. So in the end, you should evaluate what works best for your infrastructure, what would be the best legal framework to provide services. But the goal is in the end to have governance, to have annual membership, to have some resources to operate and full access to services, so develop services, provide services to the user community. Important in this context is one of the lessons learned. Maybe it's really useful to mention that keep your ministries, your countries, really ministry level engaged right from the beginning. You need them, and this is a really, as you can see, really long-term process, and having them involved helps a lot. This is what we are doing also now. So we are currently in this implementation phase, which is also supported by the Commission, and we are discussing with the Ministry in this interim phase to build Emphasis as a fully operational facility with kind of fine-tuning the business plan, developing widening membership, developing national nodes, and basically doing all those processes to get a final legal operational entity. So there are currently 12 countries involved directly on the ministry level, and additional 16 would like to be involved, but they still need this political support, or there is a scientific community interested in phenotyping, but they are in a process to become also member potentially, and they are interested in phenotyping. But what do we want to do? Objectives. So develop instrumented facilities in Europe, across Europe, and provide them for access. Then link data acquisition on the one hand, data management and modeling as a pipeline, and in the end, knowledge technology transfer. So there is a lot of technology that we developed, and providing this technology to different stakeholders, this commercial aspect plays an important role. In what we specifically do, going into some more details, what we try to do in phenotyping, this is one way how we can look at phenotyping. We try to look at entire pipelines from seeds to seeds in the end, sowing, germination processes in a vegetative, generative phase, in the end harvest, and you have many different steps. All those data go into databases, so you have your data management and modeling. This would be in basically in one slide, a phenotyping pipeline that we try to address. To give you an example, potentially it's interesting for you about seed phenotyping. So what we try to do specifically with seed phenotyping as one example. So optimizing phenotyping pipelines so that you start with your seeds that are well characterized. You know the seed physiology, you characterize your germoplast, but also seed testing for seed industry plays an important role. And there is a number of different facilities that could be used for bulk seed, individual seed, extreme detail like CT and MRI, which can be used to analyze seeds. An example here, this is a facility that we have in Jülich, basically a robot that scans single seeds. So the robot grabs a single seed. Even small seeds like Arabidopsis can be screened one by one, and you measure many different traits as an example of one of the seed phenotyping processes. But in the end, we do not do just seeds, but try to address many different aspects. What we did in the preparatory phase, basically mapping gapping. This is most likely what you are in a process to do. What is existing in Europe? What are the gaps? And we found more than 200 different facilities that are there, and we grouped them into different categories. That would be a different way of looking at phenotyping. Instead of a pipeline, you could build different categories of infrastructure, which includes, on the one hand, controlled environment facilities, where we go into controlled environment and screen plants under this well-controlled environmental conditions, greenhouses, phyto chambers, and you name it. And you screen with thousands, sometimes multiple thousands of plants in high throughput. Another aspect is field, and there we have different types of field categories. So intense field, so it's field where you have high precision equipment. You have also control the environment, for example, in phase facilities, free air CO2 enrichment facilities where you control the CO2 concentration in field experiments in a field. But we have also this lean field where you basically have field areas which do not have this high level equipment, but you can link them into a network of field sites, and you have equipment systems which are closer to breeders application. But so it allows a different way of approaching, measuring different rates. Obviously, what we also have is modeling, and here you can have different types of models, obviously, which are partly also linked to phenotyping pipelines, where you have feature extraction, which you need to model quite often, correction of, for example, spatial, temporal trends, dynamic models, modeling modeling dependence on environmental gradients, and in the end, target prediction. So there is an entire array of different models that are used in different steps of phenotyping and also resulting in some very specific goals that we aim for. Another aspect, it's information systems. So data, we talked already about data. I will skip it. So this term FAIR, it was already introduced. So this is basically everyone. I suppose every research infrastructure produces, almost everyone produces data, and we need to be fair in terms of data. There will be a more detailed session tomorrow by Cyril Pommier mostly, who will present this, how we manage data with ELIXIR. So there is a close interaction between ELIXIR and emphasis, and Cyril will present how we deal with this aspect. Then moving on, what we very briefly, a different way of looking at emphasis, how we developed. So we started in 2012 access to the user community. And we could demonstrate that there is a need to use those facilities, and those facilities were widely used for their specific aspects. That went well, and we were asked to continue with this kind of a scheme. That was complemented by the application for emphasis. So in principle, we could demonstrate services work, and then we applied for S3 and started with a preparatory phase to build a business plan. Currently, we are in this implementation phase. This is the Emphasis Go project, where we have this ministry involvement and build the next steps towards fully operational ERIC, currently with 12 different countries. Additionally, the backbone, what we have, it's the national developments. So we have many developments, substantial investments in many different countries. So there is a lot of expertise, a lot of facilities are sitting in different countries. And one of the key goals is, on the one hand, benefit from that, try to reduce basically parallel developments in different countries, but do it jointly, develop jointly new services, new facilities, new tools that are needed in the end to provide access. And additionally, we are also looking to work together with other research infrastructures, for example, like EOS Live with big project, 70 partners, 13 different research infrastructure. And this is what we continue also right now. And also widen our aspect or kind of currently phenotyping is used in agroecological research, also with 70 different partners, and provide access there. And the same is done also in FENET, where we do not provide access, but develop novel tools, next generation tools to be provided as a service in future services. Additionally, there is microbes for climate. The goal is to look at the from microbes in a soil, plant, and then environments, and across this pipeline, what happens, how to improve agriculture. And but we also look at AI for life. This is a project where we specifically try to implement AI approaches for plant phenotyping. One important aspect, the last slide. So we have currently in this project, AgroServe, there is a call for access. So it's an open call for services. They are currently in this project. There are 140 and more different services, facilities, which are available for access. So basically, you are free to apply there. The deadline is end of 23rd of October. So please have a look on the website. You find a catalog of services with all the relevant services that are available for access. The application is pretty straightforward. It's kind of a two-step application where you submit an expression of interest, and in the next step do a full proposal and obtain access as a fully free access to use those services that are available there. And as you can see, there is a quite wide range of different aspects that are addressed from different climatic zones, ecotypes, agricultural practices, up to socioeconomic studies that are possible. And as such, thanks for the attention, and I'm looking forward for the...
Thank you very much, Roland. Perfect time, 15 minutes. Okay, let's move to the next research infrastructure that I don't think need any presentation for PGR researchers. Is ELIXIR, and Andrew Smith, who's the head of the external relation of ELIXIR, should be connected online, and will tell us something more about ELIXIR and its possible interaction with GRACE. Andrew, are you there?
I am. Could I share my screen, please? Or
one moment.
Yep.
Just one moment, please.
Thanks a lot for the invitation. I'm sorry that I can't join you in person, or couldn't join at the beginning of the meeting, but as I say, thank you for the invitation. I'll try to introduce you in 15 minutes to ELIXIR, to give you an idea of why ELIXIR was set up, what types of services it provides, some of the challenges that we've faced as a result of being an ESFRI research infrastructure, but some of the opportunities that it's brought as well. So that's what I hope to do to you this morning. Hopefully everyone can hear me okay?
Yes.
So if I maybe give you some context. So ELIXIR is a life science research infrastructure, and the focus is really on data, software. So managing some of the digital assets that come from research projects nationally and from the EU. So we're trying to build a good, strong ecosystem across Europe where different partners can help to manage the data and software that come from nationally funded and EU funded research projects. If we look at why the member states of ELIXIR and ESFRI wanted to try and coordinate a data infrastructure in Europe, it's because of several challenges that we all know about. So for example, the data growth, the large volumes of data that are coming from sequencing machines or plant phenotyping or imaging. It's impossible for any one organization to be able to manage the data deluge alone. So we needed a coordinated response across Europe. And of course, the different types of technologies that are generating data now. Like I said, from image data to single cell technologies, the plant phenotyping to genomic data, we need to make sure that scientists can get value from comparing those different data sets so that they are interoperable. So the standards developed allow for data exchange between those different disciplines and domains. And it's also felt that we needed to connect all of the individual institutes across Europe, because these institutes, universities, national research institutes, they have many of their technologies in their life science labs and departments. So there isn't one single institute that generates the data. The data are being generated through thousands of labs and institutes, thousands of projects, sometimes citizen science projects as well. So for all of these reasons, the member states wanted an initiative that would help to connect, coordinate, and integrate all of these activities together to try and protect the main databases and related resources across Europe. Of course, there are in the area of health, so ELIXIR is active in life sciences generally, and we have a lot of human genomics data, which I won't mention about today, but there we have to work in a very specific way for ethical and legal reasons where data can't be transferred outside national boundaries. But I'll focus more on our plant science activities in this talk to you. So, as I've mentioned, ELIXIR is an S3 research infrastructure. We were actually added to the first S3 roadmap in 2006. So we are one of the most mature S3 research infrastructures. So as a result of being added to the S3 roadmap in 2006, we could get preparatory phase funding from the European Commission, which, as you've heard from Roland from Emphasis, that allows you to develop your business models, ideas around your legal model, and so on. And so we did this in ELIXIR from around 2007 to 2011. Then what happened is that we were prioritized by the European Council and ESFRI and given a large grant of almost 20 million euro called ELIXIR Accelerate to boost our activities. And we had one use case, one work package in that project around plant sciences. And so from as early as 2014, we've been able to connect and bring together data experts in ELIXIR in the area of plant sciences to work with other organizations and initiatives. They may need help with data management. They may need a standard developed, for example. So from the early days of ELIXIR, we've been able to work in the area of plant sciences, connecting our experts with other organizations and initiatives. And of course, we work very closely with all of the other life science S3 research infrastructures on many EU projects, which I can mention in a moment. And also, importantly, there are very strong national connections as well. So often an ELIXIR node in one country in Europe, they themselves in that country will provide services to and collaborate with the nodes of other S3 research infrastructures. So across the whole of the life sciences spectrum, there's a lot of cross collaboration at the European and national level between these S3 research infrastructures, which is really, really important. So although ELIXIR has been planned since 2006, we only formally became established legally in December 2013. This is just to give you a summary of the scale, the size, and the maturity of ELIXIR. So legally, we became established in December 2013. So at the end of this year, we'll officially become 10 years old. So you can see us as a young organization, but of course, it's also taken a long time to reach this point as well when you set up a new initiative, a new intergovernmental organization. We have 22 members, and I'll show you those in a moment. And what's different from ELIXIR to many other research infrastructures is that we use a different legal model. So we use the ELIXIR Consortium Agreement. Most other S3 research infrastructures eventually aspire and aim to use the ERIC as a legal model, and many are on a journey to create the ERIC or have already put in place an ERIC as a legal model. Well, ELIXIR, we use something different where the legal model of EMBL, the European Molecular Biology Laboratory, lends its legal personality to the ELIXIR Consortium. So we get the rights and privileges of being a full-blown intergovernmental organization. And that allowed us to be established relatively quickly, so in December 2013. So we have a strong advantage in being able to operate for a number of years now. Our model is also based on one node in each country, but those nodes themselves bring together many different institutes. So across our 22 member states, we've got over 250 institutes involved in ELIXIR, and we have a legal agreement from the ELIXIR hub to all of those institutes. So we can transfer budget to them. If they work on an EU project with us, they can join as an affiliated entity. And this took a lot of time to establish, but it's really, really important because of the way that we work. through commissioned services, and I'll mention that in a moment, because that's also something for you as a consortium to think about as well. We have 400 services, in fact, more than 400 services, as I'll explain to you in a moment. And within our network of institutes, we've got over 800 experts, data scientists, software developers, trainers, bioinformaticians, all that are active across our projects. In the ELIXIR hub, the headquarters where we do coordination, we've got over 45 full-time employees now, and that's grown from 2 or 3 members back in 2007 when we started to recruit our first employees. So we've grown quite a lot as a result of getting budget from member states and from the European Commission. The member states themselves transfer budget to ELIXIR on the basis of their net national income. So it's a bit like GDP. So the larger the country, the more they pay. And that gives us a technical budget of 35 million euro over a 5-year period. With that, we recruit staff and we fund projects in the ELIXIR nodes. That could be projects on plant sciences, so helping our scientists to work on data management projects or developing software, for example. And then we're also involved in a lot of EU projects as well. Now, these are the countries that are members of ELIXIR. We did invest a lot of time in the early days trying to build support from countries. And that's come to fruition in the fact that we're one of the larger S3 research infrastructures in the life sciences. So most of the countries that have a critical mass when it comes to data sciences are already part of ELIXIR. And this will expand in the next 2 or 3 years as well. Our funding model is based on the fact that a country transfers budget to the ELIXIR hub. And what we then do is fund projects called commission services that are carried out by our scientists in the ELIXIR nodes. And it's the ELIXIR nodes that provide the services in ELIXIR. Okay, so we don't run the services centrally from the ELIXIR hub. The services are run by our experts in the ELIXIR nodes. They're available to anyone. Anyone can use them, and I'll give you examples of them later. But also collectively, we get funding from the Research Infrastructure Programme of Horizon Europe and other parts as well, like Digital Europe as well. So collectively, we're also active in accessing EU funding. And what we try to do is help support our national nodes to get funding from their local governments as well, sometimes through national roadmap funding or responsive mode competitive grants, or using EU structural funds, or developing industry engagement schemes as well, or getting philanthropic funding from charitable foundations. So this is the funding model of ELIXIR overall, and that collectively allows our partners in the ELIXIR nodes to be able to run the services that scientists can access for free. The types of services that we provide, so obviously this includes databases, so things that you might have heard of like Ensemble or the European Genome Phenome Archive or Magnify. So some of these big databases where you can put data in and you can get data from it as well. They store a large part of the data that are generated from national and EU funded research projects. They are maintained by large teams of 40, 50 people. They have help desks. They run 24/7. So they are very, very large and successful and stable databases that can store data for the globe. We also develop software tools, but workflows, algorithms, registries to help people find software as well, to allow them to analyze data. We've got a very active network of training experts who run training courses on bioinformatics, sometimes virtual online training that people can access from our training registry. And we have a lot of effort to try and help to make data interoperable. Now, this could be data from genomics data to plant phenotyping data, trying to make them interoperable with each other. Some of our national partners provide computing, so high performance computing resources. where scientists can access that, but sometimes there's a cost if they're not from the country that is providing it. And then more and more of our national nodes are providing data management support as well to nationally funded research projects or joining EU projects to help lead the data management work package. So that gives you an idea about the types of services.
Andrew, 3 minutes.
Thank you. So what you can see here is a list of the types of services that we offer. I've mentioned many of them, which you can access for free. But we also have registries and web portals. If you want to find training courses or software or want to know where to put your data, you can use our registries and guidance documents to help find that. And for our internal partners, the main mechanism that we have are communities where they bring together experts in a particular domain. So we bring, for example, you'll hear from Cyril later tomorrow telling you, along with emphasis, about some of the activities in data management, where we connect our data management experts in plant sciences together through an ELIXIR community, and they work with external partners as well. We fund those activities, and this is what makes ELIXIR a little bit different to many other S3 life sciences, in that we have a budget from our member states which we can use to spend on projects that connect the scientists in our ELIXIR nodes. So we might want to fund our experts in ELIXIR on a plant sciences project, and we have a mechanism to do that and to fund them, known as our commission services. And of course, we also use EU projects. We're active in over 20 of them. We coordinate many large-scale ones. And I'll just give you a flavor of some of the plant science or biodiversity or agriculture focused ones as well. So projects like Bicycle, AgroServe, which Roland mentioned, and BGE for biodiversity genomics biodiversity genomics as well. We often lead some of the projects in the European Open Science Cloud, like EOSC Life, which connects all of the life science projects. And so the way that European funding works in the Research Infrastructures Programme is that a lot of the topics enable collaboration between the existing research infrastructures and new, mature, developing ones as well. So there's obviously possibilities to collaborate in future. That's where I'll finish and just leave with you some of our links to social media and where you can find out our webinars and videos, including some of the recorded training in case you want to use them. So that's where I'll finish, and hopefully that's been a useful introduction to ELIXIR for you.
Thank you very much for the thorough presentation. I kindly ask you to stay for the final roundtable. And
okay, so first of all, thank you very much for the invitation. Sorry for not being very present, but happy to have the opportunity to participate, at least from remotely. I will introduce you about METROFOOD RI, that is the research infrastructure for promoting metrology in food and nutrition, which has the purpose to provide high-level metrology services for enhancing food quality and safety and for supporting the agri-food system. We are included in the S3 roadmap since 2016 for the domain health and food, and our consortium includes 48 research institutions from 18 countries, covering both member states and associated countries, and it's under the coordination of ENEA from Italy. So we are organized with a hub and node model. Why METROFOOD RI? Because we considered to examine the so many challenges that the agri-food system is facing, and so to take into account all the aspects that are related to food quality, safety, nutrition, authenticity, sustainability, transparency, circular economy, and also the topics that we can say emerged also in the last years in relation to sustainability, but also resilience of agri-food systems that arrived so highly to the attention also with the COVID pandemic, and the importance of taking into account the so-called One Health approach, but also the agroecological transition, but with a look, with a view to all the so many aspects that are related to the food per se, and therefore also the aspects that are related to the environment, the agriculture, but on the other hand to consumers and health, so combining environmental and social aspects. And there are some aspects that, some key topics that we can identify, for which the development of new analytical methods and systems for early detection in situ and inline measurements can be really a very important point of attention, together with the promotion of harmonization and standardization. And on the other hand, we have to face the so-called digital revolution and digitalization of the agro-industrial systems by, of course, applying and making practical the FAIL approach. On the other hand, it's also important to apply inter and transdisciplinary approaches, but together with citizen sciences and co-creation approaches for involving practically all the so many actors of the agro-industrial system. And so we built up METROFOOD RI as an infrastructure, let's say, combining a physical component, which includes what we named METROSIDE, with plants and laboratories for development of new reference materials, and more than 150 facilities that are related to analytical laboratories for the characterization of foods, but also of all the matrices that are related to the agri-food. So, for instance, matrices from the environment of primary production or food contact materials and so on. And what we named the food side, covering experimental fields, farm fisheries, and plants for food processing, for testing practically all the steps that the food production chain. And then we have the electronic component of the infrastructure, where we collect and integrate and share all data that are produced by the physical components, but they are also somehow combined with external data arising from other networks with which we collaborate, and where we started also to apply IoT, blockchain technologies, and artificial intelligence for supporting indeed the digitalization of agri-food systems. So where we come from, you saw already several times today the life cycle of a research infrastructure. We started being included in the ESFRI roadmap as emerging projects, so of excellence, let's say, from the scientific point of view, but not mature enough for entering full title the roadmap. Then we have been funded by Horizon. We reapplied on 2008, the roadmap 2018, and at that time we moved to the status of project in the roadmap, so full title as research infrastructure, also mature under an implementation point of view. And currently we finished our preparatory phase on 2022, applying the first step application for the ELIXIR legal status. So we are currently on the process for the ELIXIR, and next January 1st we will start our implementation phase with the early phase implementation project funded by Horizon Europe. So what we did till now, and mostly during the preparatory phase, so of course we defined our business plan, strategies, etc., but also we defined the network of strategic scientific topics, also taking into account the policies about around the agri-food, defining our research and innovation agenda for now it's the next I would say 2 years, because it has been published on May 2022, and it's of course freely available on our website. And also we defined our service chart and mechanisms for service provision, let's say our potential users, our users are not only researchers, but also policymakers and food inspection and control agencies, food businesses from the agri-food sector, of course, and consumers and citizens. So we have different kinds of services that are differently addressed to these four types, categories of users. Here you see some examples of services that we have already provided via open calls or that are available for virtual access from our website. And what I would like to highlight is that, of course, we work also a lot for collaboration at national levels, but also with other consortia, other research infrastructures, because it's really we need that to focus on initiatives integrating different RIs, but also different networks. And so here you have a look to some projects where we are involved in that for us are very important for implementing different parts of the infrastructures and also for providing services. Some of them have been already mentioned by the previous speakers, and some of them are focused on the implementation and operation of our national nodes. I would like to give a hint particularly very quickly on the METROFOOD DT project that is funded under the National Recovery Plan. Why? Because there will be quite soon opportunities for services also in this case. And there is this open WIT database that we are implementing that I thought it could be of interest also for your community, because I see an opportunity of collaboration with Progress as well. So we are implementing a database about the multi-parametric characterization of different samples of wheat, and really focused on all the different aspects for characterizing the wheat, and it will be completely open and available from our platform. And also 3 services we are going to launch: the calls for access for Chania and virtual access. They are focused on food frauds, authenticity and traceability, and nanoparticle characterization in food and in food. And we are opening 2 new living labs for making practical this co-creation approach. One is focused on circular bioeconomy and industrial symbiosis, and the other one is the Agri-Food Fab Lab focused on sustainability. As Roland already mentioned, the other project I wanted to mention is AgroServe. Why? Because there is the call for proposal currently open, so there is still the possibility to apply for services, integrated services among the research infrastructure, so it can be a nice opportunity for participating and benefiting of the services of the 11 research infrastructures that are involved in the project. Thank you.
Can you hear me, Claudia?
Now, yes.
No. Can you hear me now?
Yes.
Okay. Thank you very much for your presentation, and please stay here because now we have the roundtable with all the other research infrastructures, and we will discuss some of the aspects you have presented here.
And sorry to interrupt. As I already mentioned to Giovanni, for METROFOOD, there will be for the roundtable Professor Gabi Mussatea from IBA, because I have to leave, but he is connected, so he will join and participate to the roundtable.
Okay, Claudia, thank you. Thank you very much, and welcome your colleague.
Hello, hello everyone. I'm here. Yes.
Okay, Gabi.
Yes, yes.
Okay, we are a bit late, so we will start quickly discussing about possible interactions between the future GRACE RI, hopefully the future GRACE RI, with the other research infrastructure. Just a couple of words, couple of considerations about why we have invited DISSCO, emphasis, ELIXIR, and so on. Oh, I'm sorry. Would you like to go on, Nigel?
Yes, I have a question. So first of all, I'd like to say thank you for 4 very informative talks. But for us, we're at the stage of deciding whether or not we will actually go through the process of applying for an RI. So the sort of questions that maybe are obvious ones to you, but we need answering are the trouble of getting money from the EU. Presumably it has to be balanced by funding from national governments to the process. And so did you have some partners drop out because they didn't have the additional national funding or consistent national support over the lifetime of going through the whole roadmap process? And then does that mean in the roadmap process, it really is focusing on talking rather than actually implementing the structure itself? So how far can you get in terms of your research objectives while you're actually going through the roadmap process? And I suppose really what we're asking is, we were informed yesterday, it could take up to 20 years to get through the roadmap process. And so what sustains the partners in their involvement over that very long time period? So if you could have some comments on that, please.
Would you like to start, Roland?
So I can start and share our experience from emphasis. So at the beginning, with when we entered the roadmap, the community was very enthusiastic, but it's a really long process. So there are many different aspects. On the one hand, you bring the community together, so you have an interaction between different countries on a scientific level. An important part is also to engage the ministry representatives as soon as possible. Based on our experience, this engagement with ministries, with representatives from the government on a national level, it also helped a lot in developing facilities, developing services on a country level. So basically the members, the countries, they obtained also resources, funding to develop something within their countries as part of the S3 roadmap, of the S3 infrastructure. And this is quite often possible once you are recognized as a infrastructure important on pan-European level. So it helps also this long-term process. There are many different aspects. Quite often it starts quite often that people are very enthusiastic about science, doing jointly science, but we will always have a number of people who will be completely engaged and completely overloaded with all this administrative stuff. You can imagine you have, I don't know, even 10 countries. Every country is different. Discussing with every country in this process, it's a really tedious, long-term process. But you obtain this long-term perspective for 10, 15, 20 years, which has a merit. So it may be useful to put this effort into that. But it's really a long-term process, and it takes a while.
Jose, would you like to tell your experience?
Well, but the thing is, I joined DISSCO not so long ago, although I've heard stories about it. It is actually hard. I remember that you mentioned that funding at the national level too. from our perspective, one thing that has shown to be useful for us is what we call the Funders Forum. And it's a huge work that we are trying to do, contacting people in the relevant ministries at the national level and bringing them together in some kind of body that it's part of the governance model of DISSCO. In fact, we have an advisory body, we have a technical advisory body, we have the partners, of course, and we have the Funders Forum. And the Funders Forum is basically a group of people with access to the ministries, some bigger levels, some lower level, and we have been engaging with them for years now during the preparatory phase. So that's a very important thing. I remember that at the beginning of this conversation, somebody asked about, okay, you're only involving scientists here, or the farmers, I would say, and what about the politicians too, right? The policymakers are the ones that are going to be implementing whatever resources, whatever outputs we provide in actual policy, conservation, characterization, restoration, etc., biodiversity. So getting involved with them from the beginning would be, I would say, it's one of the top 3 things you have to do when you're trying to put together research infrastructure. That would be important. DISSCO particularly works now how to engage, let's say, the people, the partners, right? We work on a structure of national nodes. In some countries, we have consortiums, they are together and they represent all the institutions from that particular country. In other countries like the UK or Germany, that doesn't happen. They are big institutions, they want to operate on their own. But we have been working through a network, let's say, that has representatives in each of the countries, and they work at the national level, at the local level with other institutions. So we have to basically work with the national nodes, is how we call them, and then they engage with the rest of them. That being said, what actually I believe keeps us engaged is the actual results. I mean, despite the ERICs, despite the research infrastructure and everything, the actual truth is that we need these developments to happen, right? Because, I mean, researchers are asking for them. So we have been working somehow without all this infrastructure and all this funding and everything on getting together. I'm talking, of course, about natural history collections for some time already. We have already developed. I wanted to focus my presentation, as you could see, not so much about what the history of DISSCO has been, but rather on the actual things that we're trying to develop. And those are the ones that are keeping people engaged. The fact that we know, okay, we have an actual design of a digital standard specimen that we will be able to, that keeps people engaged for a longer time, I would say.
Okay, thank you, Jose. Andrew, are you still there?
Yes, yeah. So ELIXIR, for us, the process has been very successful, and we would absolutely go back and do it all again. It has been successful, but it has come with some challenges as well, which I have to be honest with. So although it's been worth it, it has taken a long time. And sometimes it feels like you're pushing a rock up a hill. You take 1 step forwards and then 2 steps back. And if you're building a sustainable research infrastructure, it will always be like that. And for me, if you're trying to decide whether you should go forward, then you really need to be clear about what it is that you're trying to build. If you just want to do science, just then do a research project. Don't try to create a research infrastructure. Just get funding for a pan-European research project for 3, 4, 5 years, or continue to do that. If you're trying to build something that connects institutes, countries through legal agreements across borders, naturally it will take a long time. I think you also need to have strong champions, not just highly visible scientists, but particularly those that have contacts with ministries. And for me, this is the key. And if you want to use something as a barometer to try and determine whether you should go forward or not, you need to try and get a feeling for how supportive the member states are. Yeah. In my opinion, if after 10 years, you only have 3 or 4 countries that are a member, it probably hasn't been worthwhile. If you've been able to bring together many more countries than that, then it probably has been worthwhile. And it's usually the ministries, the research funders that you need to build those links with. And what they will be looking for will be to try and see how your infrastructure solves their problems. They will be looking to see how what your infrastructure does offers something different to what's already there. And the challenge fundamentally that we face, all of us, is that the budget set aside in member states and at the European Union for research infrastructures hasn't grown at the rate at which the number of new research infrastructures has grown. So although there's EU funding and there's national funding for research infrastructures, you need to be very clear about why your infrastructure needs that investment, how it's doing something different, but complementary to other infrastructures as well. Because it's impossible for any one country to join all of these infrastructures. They need to make decisions on how important your infrastructure is for their science, for their bioeconomy, for example. So you need to really be very clear about what you are offering, how that's different, and how you need a pan-European approach. Now, in ELIXIR, this journey has been worth it for us because our partners have been able to receive national funding that they probably wouldn't have got otherwise. We've been able to receive European funding that we wouldn't have got otherwise. Therefore, we've had the effect of being able to coordinate, integrate, help sustain these databases and the scientists and data management experts running them as well. But you need to be realistic. It's not a scientific research project that you're doing. You're going to be spending a long time thinking about a legal model, a long time thinking about your business model. You'll need to bring experts in to help you do that. The existing research infrastructures can share advice. We've all made mistakes. We can share with you what has gone wrong. But you need to go into it with your eyes open, that you're not doing a scientific research project. You're building an infrastructure, an intergovernmental organization, a new ERIC. And with that, you need to think about the funding models, the finance, the legal agreements. So that's perhaps where I'll finish. But I will say for ELIXIR, it has been worthwhile and we would do everything again.
Thank you. Thank you very much. And is there anyone from METROFOOD that
Sorry,
wants to comment?
Giovanni, maybe there's a Gabi from
Yeah,
METROFOOD
I'm here also.
could
Yeah,
say
I could
something
say,
about- Okay.
I could also say a few words. I totally agree with what was previous mentioned, and if I should say in one word to characterize this process in developing a research infrastructure, challenging, I think is the proper word. As it was mentioned, we also work with national nodes. We try to develop national nodes, including several types of organization. For example, in Romania, we managed to access also some structural funds in order to build our national nodes. So in big lines, more or less, we are facing the same aspect like the other older infrastructures, let's say.
Okay, thank you very much. I have are there any other questions from the floor? Yes, Nick Vangeluwe.
Thank you, Giovanni. So Nick from Euroseeds, the European Seed Association, representing the private breeders. And actually, it's really great, this research infrastructure that you propose. I think if you look at the big trend, data is the next Baustoffe, the thing to continue, build, let's say, a better future with better crops, etc. Now I would like to bring, let's say, legal aspects. So at the end of December, the COP15 came together to discuss a new, let's say, global biodiversity framework. And there they raised the points of benefit sharing obligations for DSI, digital sequence information, whatever that might be, because it's not defined. But I think you can think about it as data. So how are you as a research infrastructure going to deal with fulfilling these obligations regarding benefit sharing of data related, of course, to genetic resources in that matter.
So this is a question for Grace or for all infrastructure?
I think for the data, yeah, related. So it relates to plant phenotyping. It relates to genotyping data. Any kind of, well, it's not specified yet. So just wondering, how do you look at this as a structure? Because for the Grace RI, we will be dealing with plant genetic resources and all the data that is related to that. So. And for end users, for breeders, that will be a very important aspect. I don't want to create a big discussion about the DSI or the global biodiversity framework as such, but it's a reality that we need to take into consideration, I think.
Is this an issue for DISSCO?
Well, I think that, like many of the things that we actually discussed today, this is going to bring us to the FAIR concept, I think. In my presentation particularly, I have been talking about one-stop research infrastructure, digital research infrastructure. And when we were talking about that, we were talking about services as well. It doesn't make any sense to do anything like that unless you make it open, you make it, available for others to do. You might obviously be familiar with the concept of open science. FAIR is open science, findable. You can find it accessible. You have permission to access it. Interoperable, you can use it because you are interoperable with other institutions, and reusable. You can use it again for other things. So that effort that we have mentioned during our presentations of all these research infrastructures, trying to make things fair, has to do exactly with that. FAIR is making things, making the data available, and so that it benefits everybody, basically, in the research community.
Maybe to add from the phenotyping perspective, so I fully agree, data have to be fair and accessible for the community. There is the European Open Science Cloud. There are many developments pushed from the Commission also to make those data fair, accessible, usable. From our perspective, there are in the end many different aspects. So what we quite often try to do, not to just have this big picture and try to develop standards, which we also do, like MyAPI, so minimum information for plant phenotyping experiments. There are many ontologies. There are many different approaches, bottom up, top down. What's quite important for us is that those data are practically, they have some practical use, because we also have an experimental facility. In the end, when you do an experiment, you would like to see, you have an experiment, thousands of plants, you would like to see in real time what happens with your experiments. Is it running? Is everything still okay? And this is works only with when you have your data, when your data are accessible, and when you get this real-time view what's happening there. This is something where you quite often get also the scientists. There is a value in data also for you, for this day-to-day business, for your practical experiments. Quite often, talking just among us, when you talk with scientists and talk about them, you need to make those data available, accessible, and upload those in repositories. And they say that that's quite happens quite often. This is additional work. I don't see the value. And it's also an important part of our job as a research infrastructure to demonstrate, to discuss with those people there is a value. We are quite often selling something towards the future. What you also mentioned, that data will be permanently needed, and it is a large value in data. And again, to our phenotyping approach, we will never be able to do all the required extract the required knowledge based on experiments. So we will have to make models developed based on data that are accessible and close the gaps with experiments, because those experiments are extremely expensive. And having more targeted approach, where you use your data, develop your models, and do something that's targeted, will bring us ahead. So as such, yes, data are important, and it's an important part of the entire S3 world.
If I may, I would like to build on this concept as well. There's an important question here, and it's prioritization. Is that a word? Right? Priorities, okay, whatever. Let me give you an example from the DISSCO experience. as I said before, one of the main things we're trying to do is digitize specimens. We have 2 billion in Europe, but there's no way we will do that before, I don't know, next century or something, right? So we have to prioritize because we have limited money and probably unlimited needs. So we have to match those two. The way we're doing it, so you can actually have an idea also how to keep people engaged, is that we're trying to find the most useful effort possible. So we're prioritizing those things that we actually need. Example, it's not only that we have been working, for instance, in creating a set of guidelines in DISSCO, so we can actually explain to researchers how the digitization workflow operates. It's not only that we have been working on a cost book for digitization, so we can actually have an idea how much it is. By the way, herbarium samples are way cheaper than the others, so lucky. But it's also a matter of which specimens we digitize first, right? So, for instance, a good example of how we prioritize is we have been working with one of the complementary projects of DISSCO. It's called Synthesis. And in Synthesis, we open a number of calls during the last years, inviting people to make the case for what they believe, researchers, what they believe should be digitized. So we made sure that the things that were digitized were the things that were needed, basically. So I think that there's something to, also to have in mind, right? Prioritization is important, because as long as you continue creating the money that you have, creating useful, necessary content for your stakeholders, we believe you can still keep alive, basically.
Okay, thank you.
Okay, so for GRACE, but I think for other infrastructures as well, in general for funded projects, there are two main requirements, and they are legal requirements, and we cannot work around them. From one side, we have publicly funded data have to be fair, open, accessible, and that's a given. On the other side, there is a requirement for benefit sharing from the use of genetic resources, and also now increasingly of data linked to them. And that's the second requirement. And I don't think these two are contradictory. Actually, the more fair you make the data, the more the private sector will be able to use them. You have to devise a system to track the benefits that are coming out of the use of these publicly funded data. That's going to be the tough part, and a system to then convince the private sector, but I think that will be the easy part, to share the benefits with the generators of the resources and of the data. So I don't think this is contradictory. The main difficulty I see there is the tracking of the use.
And I guess this could be done by interlinking and connecting all the different research infrastructures which keep the information. Am I right, Andrew? Could you give your point of view about this?
Yeah, so maybe a quick reflection on this. So obviously ELIXIR's initiative to try and connect, coordinate, sustain some of these databases and linked resources in Europe. So right now you have databases like the European Nucleotide Archive, Ensemble Plants, for example, and you've got scientists all around the world that put the genetic data into those and get data from them as well. So there's already resources that are being used in this genomic area. But I think it is important to think globally for benefits. And as an infrastructure, we do try to collaborate with institutes and organizations in Latin America, Africa, and other world regions. So we have a scheme where we use budget from our member states to allow collaboration between European partners and scientists in those countries. Often that is around data management. So helping to try and train and upskill and build capacity on data management, because we have registries and we have toolkits that everyone around the world can use to help build their skills when it comes to data management, or the standards that we develop are maintained by the community and can be used and adopted by scientists everywhere. So I think it is important, the whole topic of global benefit sharing, and as infrastructures, there are things that we could, or that we can and should be doing to help make this equitable.
Any comments from METROFOOD?
Not really. We are not dealing mostly with this type of, but we are using it also, all the FAIR principles and open science, and we are developing databases for sharing our data, of course.
Okay, any more questions from the floor? There is a hand raised, and since we are 20 minutes past the end of the coffee break, I would take this last question and then close the round table, if you agree. Lisa Lycke-Stevenson.
Thank you. Thank you very much for some very interesting presentations, and to see your roadmaps, to see how you have where you started and where you are now. I have two questions, which I think has been discussed in the meeting and in the corridors here. One thing is the speed. You all have had 4 years where you've had a good time to plan, to think, to consolidate, and come out strong. So we have been in this project for 9 months, and there might be a call next year. So to ask, based on your experiences, if you were to, after just 9 months and then maybe a year, and what best advices can you give with that for going for such a short process, or would you rather wait for the next one? You sit with the experiences. The other one, which is very much in the corridors, is about funding. How much funding are you getting from the EU? I mean, are the percentages? How has it gone over the years? And how did you succeed with this? There is a true fear that there's just an amount given at the ministries for plant genetic resources, and if some of those should be given to an infrastructure network, then it might go from the actual activities today. So how have you sort of crunched that nut? Thank you.
Roland, would you like to try to answer to both the questions?
Yeah.
Bit complicated.
It is a little complicated to provide an advice how to move on. To a certain extent, Andy addressed that. So it's really important to think how do we want to continue as a community? Is it really worth to invest 10 years of time to develop this big, heavy tankard, or something that's sometimes big infrastructure? It might be also useful to have something flexible to adapt to different needs, but this is something that you would need to find out as a community. And it's good to have this project that you have now, and but you apparently have some time issue that you would need to apply for the next roadmap. Not completely sure what to advise, but keeping the discussion going, it's important. I can see the value also in having, because it was already introduced, there are many different gene banks across Europe, and linking them, it is important. From my feeling, I think that it would not work with projects. Having just projects and trying to link a pan-European, developing a pan-European network of gene banks. So the research infrastructure approach might be actually a pretty good approach to provide this, to have this pan-European approach to link the gene banks. In the end, how you do this in a formal way, in the end, there are many different approaches. There are also some light research infrastructures which have a very agile small offices, but the basic idea is to have national development that are linked in some way. And also in terms of writing this application, I think it's like a proposal, writing a proposal. So I would not see this kind of this writing process as an issue, particularly for you as a plant genetic resources. This is important. This is needed in linking this. That's important. And I'm pretty sure that also the Commission, ESTRI, would consider this as important. In the end, you have also the design project funded, which is a pretty good sign that also they want you to do the next step. But how to address that? I think looking also at the roundtable. So we would be right now talking also for other colleagues, but you are smiling probably now with your. So we are happy to stay with you in contact and discuss with you and provide. Basically, when you learn from the experience that we had, for us it takes probably 10 years to get to an operational infrastructure. When we can shorten your time to get there by a couple of years, that would be also kind of rewarding for us as well. So happy to help you, happy to be involved and discuss with you.
Well, I'm not going to say a lot of things different from Roland, obviously. But I do agree with him that research infrastructures are important rather than projects, because if only because the European Commission has made very clear that it will talk with research infrastructures. So you need to become an actor that is heard, whose voice is heard at the European Commission, period. That would be the first thing. Advisors, okay, well, grandpa advisors I can give you. We spent in DISSCO over 3 years getting ready. I explained that before. We had a first stage, DISSCO Prepare, we called it. During those over 3 years, we tried to get ready in terms of science, in terms of data science, in terms of technology, governance, financial matters. So I would not be in a hurry. It takes time to get ready, actually. Important thing, focus, I would say, get your governance ready. Think about it. What's more important for the purposes of your future infrastructure? The same with the funding, the same with the technical aspects, same with the science aspects, etc. Take your time to do that. That will be the first one. Second, differentiation. We have heard that before too. As long as you provide something different and you're able to justify why you're doing what you're doing, you should be fine. So also put your energy on that too. I would also talk about interoperability. I've been talking about that the whole time, but the thing is that the more you are able to work with other research infrastructures, probably the better. So you have to work on that. And the fine thing about progress is that you are in the right moment right now. You said before about funding. I'm not scared about funding. I'm scared about the monumental work that we have to do behind the scene, in the background to develop all this technical infrastructure so we can eliminate gaps and we can connect, I don't know, in your case, we can connect the herbarium samples with seed banks, which sometimes are not connected, so sometimes you update something, the other thing stays the way it is. Sometimes the content management systems, they don't work together. We need to make them work together. And that entails a lot of work. I mean, one of my colleagues, Shahid Islam, is in the conversation. He might tell you stories about it. It's really difficult. So, as I said, differentiation, interoperability with others, and clear work of focus on what it is that you can actually, that you really need in order to continue the building. Don't take any rush on this, I would say.
There is Drew Smith online.
Sure thing. Just a quick reflection from me. So yeah, it's very hard to say whether you should or shouldn't. That's something that you need to assess yourselves. But what I would say is that in terms of the time frame, if you do go forward, it's much harder for research infrastructure to build momentum and have the support of the experts who need to prepare the application if the current EU project that they're working on is ended. Now, and we know this from experience of looking across all of the ESFRI research infrastructures, once your EU funding is ended and before you exist as a legal entity, that is a really difficult period because everyone's gone back to doing their day job, teaching or running the seed bank or whatever it is that they're doing. So try to make the most of the EU funding that you have to move forward. So I would try to use that as something that influences your thinking. And of course, now the process for submitting on the S3 roadmap is different at this time than what it was in 2006 when ELIXIR went forward. But what's still true is that we knew we had the support from many, many, many countries, many governments and ministries of science. So we felt comfortable to move forward, even knowing it was going to be a challenge. So what I would say is that if you're thinking of going from your design phase to your preparatory phase funding and applying for the S3 roadmap, if you've got an indication that's the ministries of science or agriculture, whichever ministries you're in contact with, are supportive, and you've got enough countries to maintain that support, then for me, that's quite an important factor. If you've already got a feeling that there's no interest from the ministries, it's going to be a real uphill battle.
Thank you, Andrew. So, as we say in Italy, you have to throw your heart behind the obstacle and start thinking about this roadmap. So, if I would close this roundtable here, thanking all the speakers and proposing that we hold perhaps a more extensive